Welcome back. I hope at least most of you got to go outside to eat your lunch because it is amazing out there, but I hear it's terrible now. Storms, everything, but really beautiful day out there in DC. And you will be happy that you're back though, because these three presenters are going to knock your socks off, and I think it really relates to a lot of the points that came up earlier today. So this is showcasing impact with emerging platforms. So let's get right into it. We are going to start out with Meghan McCarthy. She's a program lead of 3D printing and biovisualization. You're excited already, right? From the National Institutes of Health.
All right, great. So here we are. So good morning, everyone, or good afternoon. I hope you're all awake after lunch. You don't get that post-lunch slump. So as Liz said, I am with the National Institutes of Health. This is our fun organizational slide. I am with the National Institute of Allergy and Infectious Diseases under the Office of Cyber Infrastructure and Computational Biology, which is essentially our fancy term for the IT department, and we are the scientific branch of that. But our branch, Bioinformatics and Computational Biosciences branch, does much more. We have PhD researchers, software developers, and we create custom tools and platforms that help researchers at NIH and the global public make better use of, better access to and analyze bioinformatics and clinical data. So in 2014, we launched the NIH 3D Print Exchange, which is an online repository for 3D printable models related to bioscience and medicine. We also host some very cool tools that I'm going to tell you about. So what I'd like to think, so we've been kind of, we've known the digitization program at the Smithsonian for a couple of years now and done some work with them. But what I like to think of, we're kind of the opposite end of this spectrum, whereas they're taking these great 2D and 3D objects and scanning them into digital collections that people can access. We are taking digital representations of molecular models and medical image scans and turning them into 3D printable models. So kind of taking it from the digital to the physical world, or as one collaborator said, atoms to bits and bits to atoms. And in the last year, we've been working more with virtual and augmented reality to embrace that whole spectrum of 3D technologies. Oh, sorry. So we started with 3D printing in 2007 for the purposes of better molecular visualization and understanding. So our current branch chief and structural biologist, Daryl Hurt, started using a color 3D printer to create these models that researchers had been looking at on a 2D screen with or without 3D glasses, and there's only so much you can gain from that. And when he printed them out and handed them to the researchers, even people who had worked on a particular molecule or disease for 10 or 15 years saw something that they hadn't seen before. So that was really exciting, and that's sort of what spurred us on to create the exchange so that we could share those. And so this picture was taken in June. This is our foray into virtual reality. On the left is my colleague, Phil Cruz. And you may recognize the gentleman on the right in the blue sweater. That's Bill Gates, who visited NIAID's Vaccine Research Center. And we did our little demo for him, showing how we are taking these molecular models and bringing them into a virtual reality environment and allowing researchers to actually go inside them. And this molecular spelunking term was coined by a research collaborator at the Institute for Child Health and Human Development, where he went, you're scaled down, so it's the opposite of 3D printing, bringing this small object to the scale of life. You're actually being scaled down to get inside of all the nooks and crannies in these really complex structures. And he said it felt like being in a cave. He was actually able to go into this binding pocket where a drug would bind and look all around. And his hobby is spelunking, cave exploring. So he said it was like a molecular spelunking. So we thought that was a great way to kind of get that across. And that's been extremely useful. The VR, AR, and the 3D printing, we're really happy that the researchers have latched on to that and that there is scientific value in it. So we're also involved in some projects for medical 3D modeling, and we host that kind of content on our website. The 3D printing in the medical area has really taken off in pediatric cardiology and cardiology in general, because those structures lend themselves really well to being a 3D print, where the anatomy is so key to its function. And we're also trying to facilitate the open source hardware movement, particularly in relation to laboratory equipment, where the ability to customize tools and apparatus for research, being able to customize it, download it, and then print it out on demand as you need it is extremely cost efficient, time efficient, and it allows researchers to design things as they would want to be designed, and leading to better scientific reproducibility. So I'll tell you a little bit about, since we're talking about digital collections, sort of the back end of what we work with. We built a site with completely open source tools. We use the Drupal content management system. We have a whole bunch of scripts written in Python, PHP. Our pipelines, which I'll tell you about, use UCSF Chimera, which is an open source molecular modeling software from the University of California, San Francisco, and the Blender 3D modeling program. So we don't have Maya or anything really fancy, but so we're going on a budget, but all our scripts are open source. So here's an example of one of these pipelines, or two of them actually. So at the top, that DCM is a DICOM file. That's what comes out of an MRI or a CT scan. And then on the bottom is how we work with protein structure, molecular structure data. So here's an example. So essentially these files are just atomic coordinates, just a point cloud with other information about bonds. This particular molecule, this is a model of a trimer of the insulin molecule. It's just a text file. It's almost 8,000 lines, these atomic coordinates. Our pipeline outputs 8 different variations on that structure automatically based on that text file. And these different structures, there's kind of 2 monochrome if you've just got a monochrome printer. If you've got a color printer, you can print them out with the different domains and surface coloring that are valuable to better understanding. So as I said, from that one text file, we generate 32 new and unique data files. So there's 2 STL, those are our blue ones, 6 WRL, which are the color. And then for each of those, there are 8 X3D files. X3D files can be printed, but they can also be used in other ways for visualization. The STL and VRML get zipped up into a zip file that can also be downloaded. And then the X3D, because we allow for an interactive 3D viewer in the browser, we use the X3D binary format, which allows for faster and progressive loading. And then, of course, there's 8 thumbnail images. So rather than a content management system, we've actually created a content generation system. The reason why this is important is that we have lowered the adoption barrier for people to be able to create these types of 3D models for printing and visualization. So any user who makes a free account can log in, use this quick submit tool, enter a database accession code from either the protein databank, electron microscopy databank, or PubChem. We reach out, grab the structure, but we also grab metadata from the header file. And we're able to take that metadata and insert it into the database. So literally by just entering the 5MT3, we've created this whole, brought all of that into the database, and it's completely automatically curated and makes our life really easy. So I am going to get into these formats and metadata, but I'm hoping that you guys will find this useful or interesting. So STL is sort of the status quo in 3D printing. It just contains geometry. That's it. So you have no other information about it. I kind of analogize it sort of like an audio cassette where you just have the audio and then there's a label on the outside. If you tape over that audio, you don't know. Same with the STL if you version it. If you change the label, no idea. So what we want to be able to do is use other file formats that allow for embedding metadata, but more importantly, human-readable metadata. And the file formats that we see as really being impactful for the future are 3MF, which is the 3D manufacturing format that came out about 2 years ago, which is based on X3D. And both of those are open source formats, so they're extensible, they're royalty-free, they can contain color and texture information, and the X3D also contains scene info, so it can be used in animations as well. And let's see. So I'm going to focus mostly on X3D because that's what we use, and I'll talk a little bit about the portability of it. So that metadata, and I think we can get into some of this in the panel, so I'm going to kind of skip through this, but being able to add that to the geometry so that this information about the structure is attached to the geometry and travels with it is important for content creators and designers so they can license and give attribution. It's important for scientists so they can include information about the raw source data, a publication that's associated with it, a database identifier. For medical models, it's going to be important moving forward in order for these to become part of the patient's permanent health record, and the 3D models are actually going to be the DICOM standard, that file format. They've just now voted to incorporate a couple different 3D file formats into the DICOM, so that when you go to if you get an MRI or a CT and you ask for the CD to take with you, that'll be an actual 3D model with it if they've created one. And then the other key part for 3D printing is the ability to embed printer parameters so that rather than you've given me a file and it should be a certain size and look a certain way, you can include that, you can include the resolution so that the prints actually end up being printed the way they were intended, which is important for in particular structural integrity. The metadata can also be parsed to make these shareable and interoperable across different platforms using web APIs. And then we also want the data to be accessible and transferable. So particularly in the case of a medical model, if a radiologist or a technician has segmented out that data from an image stack from an MRI, if they can just use that one file in various different ways, there's much more trustworthiness to the integrity and the accuracy of that data and who it applies to. So the ability to use something in a 3D printer, in a browser, in a mobile app, and in a VR or AR headset. And for that reason, we really like the X3D file format. And as a disclaimer, I do advise the Web3D Consortium that manages the ISO standard for that, but they are a nonprofit, and I don't get anything from them. But I do think it's a great opportunity. So I'll leave you with the fact is that I think open source formats are going to be really key moving forward, and we need to try and anticipate as best we can how these kinds of digital models will be used and can be used in the future. Models that are being shared for the purposes of 3D printing could easily be put into a video game. Is that okay? Did the creator intend that? Does this affect how we share things? How do we track things and track versions? And then also this metadata is important as far as digital collections go in helping us to be able to organize and curate them. And then we've also, as I've shown, used the metadata to drive development of some of those tools. So just my quick acknowledgements. Our current team is very small, but we've had a lot of great contributors and collaborators along the way. Of course, want to recognize the Laser Cowboys and everyone at the Smithsonian who we worked on other projects with. So thank you. So just next up is Paul Franzen, who is a data scientist for the Smithsonian.
All right, I'm very excited to be here today to talk a little bit about what we're doing to scale up data science in a museum setting. I start a lot of my talks with this picture, but I think it's also relevant for this talk. So here are two museum drawers taken from the Natural History Museum just across the mall here. And to someone who is an entomologist, like I was trained as an entomologist, I see kind of like a dizzying array of different function and form among these insects. To many of you, they might look like dusty drawers from a cabinet, which is also partly true. But then if I put on my hat as a data scientist, I see the potential of a lot of different data here. So each of these insects has a pin with it that contains a label, and that label has information about geography, the time that the insect was collected, by whom the insect was collected, and so on. And so kind of museums have a wealth of information just like this that can be harnessed in a data science setting. And so these are just two drawers from a collection that has 35 million insects in it. And that collection is just one collection in a museum with several different departments. And that museum is just one museum in an institution with 19 museums. And so as you can see here, there's a tremendous potential to harness those data and be able to gain insights through them in a data science setting. And so the Smithsonian also kind of has some unique challenges. It's a unique place with unique challenges. And I'm going to kind of form my talk around these three different areas. So first, there's a diversity of locations. The Smithsonian is spread out. Second, people generate different types of data for different questions. So there's this kind of big increase in heterogeneous data, and it is coming faster and faster than ever before. And then lastly, because it's unique, there's kind of a lack of purpose-built software tooling to be able to gather the insights that we want from these data. But rather than look at these as challenges, we look at them as opportunities. And I think we have this tremendous opportunity to kind of solve these problems and use these as focal areas for our further research. And so kind of noticing the opportunity, the Office of the Chief Information Officer, which is basically our IT department, hired a few of us data scientists. Rebecca Deco and myself are data scientists in the newly formed Data Science Lab. And we're housed in research computing, which is led by Beth Stern, which is in the Office of the Chief Information Officer, which is led by Darren Burba, who gave the introduction today. And so now I'll go through kind of each of these opportunities and what we're doing in each of these areas. So first off, there's a diversity of locations, and I'm sorry that the type is just a little bit small here. But the Smithsonian is made up of 19 museums, 9 research centers, and a zoo, and then various archives and libraries. And so this varies from all these things that you see on the mall today while you're walking around to the Smithsonian Tropical Research Institute in Panama, all the way to Front Royal, Virginia, where we have endangered species at the Conservation Biology Institute. And so in order to understand and gain understanding of the types of data these people are generating, we've gone out and visited them. And we've sat down, talked with staff and researchers, and really learned a lot from what they had to say and what their needs were. And one thing that we did learn while we're out there is that a lot of people have a lot of data. And they're all doing different things with those data. And most of it is digital in form. So this comes from museum collections, from research expeditions, etc. And so here's just a list of some of the data that we encounter at the Smithsonian. We have images from mass digitization and 3D projects, which is mostly what we're talking about in this workshop the next couple days. There's museum collection databases, so all of the digital data surrounding the collections. There are data being generated all the time from sensors from ecological projects, such as those at the Ecological Research Center in Edgewater, Maryland. And then we're also creating a lot of gene sequences. And there's even more than that. There's sound, video, all sorts of different things. And so data these days has become something that is a buzzword a little bit. I even hear commercials on NPR about, come, there's big data. And one of the reasons that this data has become so important is not only do we have this capacity to gather data like we never have before, but we also have new techniques for analyzing those data. And one area that's gathered a lot of excitement is machine learning, which the keynote addressed today. And I guess this is the Bill Gates session. We all have pictures of Bill Gates. But back in 2004, he said, if you invent a breakthrough in artificial intelligence so machines can learn, that is worth 10 Microsofts. And Microsoft was maybe the most valuable company in the world at that point, probably pretty close, if not. And so that's quite the statement. And indeed, in the last decade or so, there has been a lot of research and a lot of progress in the area of artificial intelligence and machine learning. And kind of the most exciting development among these is this technique called deep learning. So the ideas surrounding deep learning have been around for a couple decades now. However, we're just now coming to the point where we have enough data, the appropriate computational infrastructure, to be able to kind of use these algorithms to gather insights. And so the basic thrust behind deep learning is that you have something like a neural network, and you give raw inputs and to train this network, so you have some sort of data that you want it to learn from. It learns automatically what the differences between your inputs are, and then it can output a probability for any unknown that it contains the thing that you're interested in. So for example, it's used in image recognition. This is one area where it has done really well. So say you have a network that you've trained with images of bears, bees, and birds, and you decide to take some unrelated bird and put it through, you'll end up with some probability that that image is a bird. And these techniques are really powerful and are driving a lot of the technological advances that we see today. So if any of you have an Amazon Echo, that's all powered by kind of these deep learning algorithms. And you may not be that impressed, but if you would have tried voice transcription 10 years ago, we've made a lot of big strides. We can also do things like pick out individual pieces within an image and say, oh, this is a boy, or this is a dog, et cetera. And so the Smithsonian have been generating a lot of images with their digitization project. And so we started a collaboration with NVIDIA and the Natural History Museum and the folks at the Digitization Program Office and the Data Science Lab to try and bring some deep learning techniques and see what we could learn about some of the collections that we have here. And I'll just describe briefly two projects and kind of how they worked out. So the first is, here's two herbarium sheets. They're basically 2D, and you probably saw some information about the conveyor belt that they're rapidly digitizing these. And so years ago, collectors would sometimes, to preserve their plants, either dip them in mercury or paint them with mercury, and this kept pests out. And as a result, you get this characteristic mercury staining on some of the herbarium sheets. And so we want to know, can you train an algorithm to be able to tell if an herbarium sheet has mercury staining on it or not? This isn't something that's generally in metadata, but it could be something that's useful to know. And so since it wasn't in metadata, we had to build out our training set. So we sat on a cold January day, a bunch of us in a room, and went through thousands of images to find images that had been contaminated and some that had been clean. And so we generated a neural network, and here are some of the results. I won't go too in depth with these graphs, but basically these images on top all should be unstained. You can see, almost 1,000 were classified as unstained, where we only, about 46 were wrong. And the same thing on the bottom, they should have been stained. And we get about a 90% accuracy rate, with the ones in the middle being actually really hard. Maybe they just have a little bit of staining, or maybe there's a little bit of mold or some other contamination on the sheet. And so if you discard kind of the 10% hardest images, all of a sudden your accuracy rate jumps up to almost 95%. And so this is a really powerful method. The next thing we wanted to try to see if we could discriminate between families of plants. And so here's two families. One's called the spike moss, one's called the club moss. And to the untrained eye, they look basically identical. Even to the trained eye, you have to have some sort of expertise and look at them closely in order to discern between the two. And so we wanted to see, well, can we train an algorithm to do it? And so we tried it, and it worked really well. So just right off the bat, we have a 95% accuracy if you discard the 10 most difficult, which if you look at those images, often they're just something with a little envelope in it or something. You're all of a sudden almost at 99% accuracy. And according to the curator there, he said that's probably even superhuman already, is that these algorithms can discern these things better than even a trained curator can. And so that's not, I should step back and say that's not to say that these algorithms will ever replace curators. What they do is very important, and that's not our goal here. We want to make their lives better. And so we decided to write up these two results, and happy to say recently got accepted to the Biodiversity Data Journal last Friday, so that should be coming out in print within a couple weeks or so. Aside from images, we also have a lot of other digital data, such as genome sequences. So gene sequences are a type of digital data. It's a type of digitization of museum objects, even, of the genotype of those objects. And so we focus on those in our data science lab as well. So we can do things like solve old mysteries about Tasmanian devil pathogens, as well as help save endangered species. And so then kind of my last little piece is the software tooling. We have access and run a high-performance computing cluster. And so we work to try to get algorithms running in parallel across that. We have advanced GPU technologies for our deep learning methods and a lot of CPU power to solve the rest. We're working with industry partners to implement some of these algorithms in the cloud so that they can be scalable and repeatable. And lastly, we're working with researchers to try and manage their data, because the data is no good if it's just on a hard drive underneath someone's desk. So we're doing our best to understand the standards that we need to keep and the metadata we need to gather in order to make sure that everybody's data is somewhere safe, and also, important for data scientists, somewhere computable. Because if we want to make some of these massive, grand ideas out of the museum data, we need to have it all in one place and somewhere that we can make connections. And with that, I'd like to thank the collaborators on this project, and thank you for listening. So next we have Rolf Mueller from the Department of Mechanical Engineering from Virginia Tech.
Okay, thank you very much, and let me see. So I want to inject a little bit of an engineering perspective here. And one key challenge that engineers face is that the machines that we build, they get more and more complex, so there's nothing special. Here's a car engine from a passenger car, and you see already how many different components that has, and it gets worse, right, if you think how important, say, the car engine is to society, right? So you have implications, I don't know if you can see that, but all the raw materials for the car, right, they come from all over the world. The way we build a car engine, it affects job, right? Who has a good job, who has a bad job, who has no job, right? If you change the car engine, that will change transportation, right? Pieces being taken all around the world, oops. Raw materials, pollution, city planning, our social life, how to deal with waste and recycling, right? That all depends on how we build these machines. So the goal or the task for the engineers is to look at this and say, okay, so how can I design, solve this really complex, multi-dimensional design problem, right? And in the end, this is again about the physical world. Right? So it's stuff so we need to design a good solution, and it has to work in the physical world. Right? How can we find that good solution in the physical world? But the most straightforward thing is to do experiments in the physical world. Right? So for example, if you want to know how a large passenger airplane behaves when you change the load, right? You need to take a large passenger airplane and you need to outfit it with these tanks so that you can shift the load in the passenger airplane, and then you will get if you get a lot of flying time on the airplane and you burn through a lot of aircraft fuel, right, then you get definite answers, right? The problem is that is obviously something that you can do only very selectively, right? On the other hand, you could say, okay, I can try a lot of different versions, say, if I take a very simple computer program, right, and I can change that computer program and suddenly I can test billions of versions of that computer program and see which one behaves best. But that is limited to having really a very short, easy piece of code, right? And of course, I can then say, okay, maybe I can make my computer code a little bit more involved, right, that it maybe mirrors the physical world better. But as I increase the complexity, the ability of my code to mirror the physical world, right, the amount of tests that I can do goes down again. Right? So that's the sort of the conundrum here. We can either have something very simple, we can try a lot, we can optimize over with very many tests, but it will not tell us much about the physical world, or we can really embed us right in the physical world, and then maybe we can test 3 things or some number like that. So that's where sort of the value of say what you find in the National Museum of Natural History, right? So the biodiversity evolution comes in because evolution is some kind of optimization process, right? So here's an example that people have tried to formalize that as genetic algorithms. I know there's a lot of, in the details, a lot of differences, but if you see how evolution operates and the optimization problems that engineers have to solve, in the basic goal, in the constraints that you have, in the pitfalls that you encounter, there's a lot of similarity, right? So that's a really good news, right, that you have this gigantic process of the evolution of life on Earth that has been going on for about 3.9 billion years, right? Astronomical number, or maybe not astronomical, but many, many millions or tens of millions of species, and then really astronomical numbers of individuals, right, that nobody needed to pay for, right? They just sprang up and they were tested in the physical world, and maybe their genes made it in the next generation, or maybe they didn't. Right? So that's really an enormous resource, right, where things have been tested, where the results of that testing has been documented in the species that we have currently, in the species that we have in the Smithsonian, in the fossil record, and so on. And so you can think of that whole evolutionary process, right, that has generated the tree of life. You can think of that as really a process that has created optimized systems and devices, and in that sense, engineering knowledge, right? So if you look at sort of human development, it's a bit ironic, right, that the same process has, of course, created waste, right, that you can find in the fossilized biomass, right? And so as humans, we have gotten very efficient to mine that waste, right? So maybe too efficient for our own good. Right? And so that reminds me of something, right? You get a Christmas present and you throw away the present, or you ignore the present and you just keep the wrapping, or worse, you keep the waste that was produced in making the Christmas present. Right? That's the wrong thing, right? The present here is really the evolution. Right? And I think biodiversity has a lot to offer to engineering. Right? So one example that people will know of bioinspired engineering is that people have looked at surfaces of plants. Right? So I think almost 2 decades ago, somebody came up and said, look at the leaves of the lotus plants. They are self-cleaning. Right? They have a special surface texture, and that spurred an enormous amount of papers and companies that came forward and said, hey, we can make such self-cleaning surfaces ourselves. Then a few years back, people said, oh, look at the pitcher plant, right? The plant that has these pitchers and it attracts the insects to fall into that. That has a wonderful surface, as I say, even more interesting properties than the lotus. And now you find, almost every week you find papers about that. Right? But if you imagine, right, what these two plant species, right, how do they relate to what is on all these herbarium sheets in the Smithsonian that we have been talking about on various occasions today already. Right? So really just a tiny sliver of that information. Right? So what if we could get all the other things out and we could look at what their surface properties are. And I think we have an opportunity to that, right? Because we have this convergence at the moment. We have the automation, the digitization efforts on the biological collections. Okay, that gives you a lot of digital data. That's not good enough. Then on top, there's the possibility, right, to actually analyze biological function computationally, right? So the specimens that are in the drawers here at the Smithsonian, they are dead. We cannot get them back to life. But with computational methods, we can take a look. What are these actually doing? But then you have data science, that also came up during this meeting many times already. That's motivated that we can deal with large data, where we can search for patterns in there. And then we have this progress in bioinspired engineering that we're learning better and better. How can we actually link the engineering world to the biological world? And I think if all these 4 developments, they come together very nicely, right? And they should allow us to really get engineering knowledge out from those collections, right, if we put them together right, so that we can sort of make this field of bioinspired engineering progress, right, where it's not just this hunter-gatherer thing where somebody happens to come across this one specimen and say, oh, that's fantastic what I found here, but really being able to search, being really able to look for solution and mine this knowledge just as systematically as we are mining the coal and the other sort of fossilized biomass that we are digging up. So with the last slide, maybe a little example from my own research. So I have been in the collections of the Natural History Museum for quite a bit. And what I've done, I'm interested in sensing. Right? So what I've done is I've digitized ears, so 3D models of ears from bats, right? These animals that can use the sonar system. And so having these models, that's sort of on the left-hand side, if you go one to the right, we can use computer methods and predict the acoustic properties, right? So the ear of a bat is an acoustic receiver, and so you can predict how it receives sound by just knowing the geometry, right? And then, so that would be the computational analysis of biological function. Then what we have done is we've said, okay, what are the principal components? There's about 1,200 species of bats, most of them here represented in the Smithsonian. What are the components of that variability, right? Can you explain that? Can you, and that's the cryptic picture number 3. And then what we are doing now is we are building robots, right, where we reproduce the sensing behaviors of bats, and we have some industrial partners, right, who get very promising results out, industrial and defense, I should say that, where we can get something out of that. So with that, I'll finish it, and then I think we can move on to the panel discussion part, so Liz can come back to lead us on that. So thank you very much.
Up here. Thanks so much to all of you. I'm just so fascinated by the work that all of you are doing. And so we're just gonna, we'll start out with some questions here, and I kind of wanted to start with a big picture one. And I'm actually gonna direct this first to you, Rolf, because you were just kind of talking about this, because it's a question that I had about museums and their role in the, like, as we look at digitization more broadly, what is its role in the real research ecosystem, even beyond what we've had? And I think that from listening to your talk, I feel like, I felt like there's this big hole if we don't have this data, and we don't analyze this data from across that museums are holding, that we're actually like really missing out, and that by doing that, you're really showing a new role for that. But so I'd love to hear from all of you about that, but if you could jump in there and say kind of like how is this new access to collections data in, you know, as being a professor, really changing the role of where a museum sits in that research ecosystem?
Yeah, I mean, in engineering, this bioinspired engineering, right? The idea that you can look at a biological model system and you can come up with a new idea, that is actually taking off, right? So I have this hobby that every year I go into the database of the US Patent Office and I count how many patents mention bioinspiration, biomimetic. So I have a couple of terms, and it's an exponential growth, right? So that's great. And then if you look at how many species do people work with, right? So what do they have in terms of data? Right? And you can really see that it is relatively few systems, right? Every couple of years, somebody discovers a new thing. With my bets, I'm of course always an outsider, but somebody discovers a new system, right? The lotus leaf, the butterfly wing, and then people converge on that, and you really have this outburst of creativity and publication. But then there's again a lull, and then it comes again. So I think if we had, through these digitization efforts, if we had this data on this really huge biodiversity, well, maybe the whole field of bioinspiration at the moment, I don't know by how many model systems that is being carried, but less than 100, right? And then you have millions of biological species out there, right? So I think, and then all these methods from, so just having these model systems, that's one thing. And the other thing is, again, the ability to use big data. That's also something that is very interesting, of course, in academia. People look at that a lot. And so fueling this, so just providing better model systems, but also transitioning from individual model systems to being able to search systematically through a large amount of data. I think for engineering, for these interdisciplinary areas, I think that I see that as a game changer.
Either one of the rest of you want to comment on that?
I agree. Yeah. I think it's also kind of, as someone who is trained in kind of basic science, biodiversity science, it's more kind of motivation to go out there and learn more about these different types of species, and a collection is a great place to do that because, as Rolf mentioned, there'll be something new found that this organism does this cool thing hearing-wise, but there's probably tons of that stuff that we don't know anything about. I mean, less than 1% of the species on Earth have even been studied in any detail, and so collection's a good place to start that sort of thing. Yeah.
So I'm going to start this one out with directed to you, Meghan. And you brought up future-proofing. I believe that was you. I kind of wanted to, basically, this is the metadata question. So I just want, diving more into that, as you started the project, is that something you knew from the very beginning? Is it, how did you address it? Where are the gaps? And I think that the two of you also have something to say about metadata. But I'd love you to kind of dive a little more deeply into that.
Sure. So I guess the question for us, so there's other 3D repositories like Thingiverse and PinShape and Imagine, all these places where you can find 3D printable files. But we went, this was 2013, we went and searched biology and there was like 100 things out of 100,000 in Thingiverse. So for us, the key was, well, how do we bring value to these digital assets? And that's by including all this additional information. And we're even working on that now with a focus on medical models and assistive devices to help people search for those better. So like our main users would be researchers and clinicians, but a lot of educators, and students, so to help people find, like, drill down more into what they're looking for. So that's sort of from the database perspective. And then I think as the 3MF came out, I want to say like 2 years ago, and it's only been in the last year that people have started to really use it, I think. But that was something that the 3D printing community really needed, was to be able to have those printer parameters within the file, which is important for distributed manufacturing and additive manufacturing workflows moving forward. So your jet engine part, isn't going to fall apart. But yeah, I think that's more important, and to have something, data that's archival, and lossless compression. I think that's where the data, the standards come in, which to the original question, it wasn't something that we thought about initially. We just sort of, created this project flying by the seat of our pants, and then all of these other things came to light that we're really starting to think about and getting out and encouraging the community, I hope, to think of the same things. I mean, we don't have the answers. We just kind of want to throw it out there and see what happens.
And as far as what role does metadata play in the deep learning projects that you're doing? And is that even a more emerging standard, or is it using the standards that are within the collections, or how does that work in your world?
Yeah, well, it certainly makes it a lot easier to have good metadata. So, the two simple projects that I described, one of them we had metadata associated with the images associated with the problem that we were trying to answer, which is what family do these specimens belong to, and the other we didn't have metadata about the mercury staining, and so we had to go through by hand, which was a lot of man hours to go through and classify all of those. And so, I mean, for me, the more metadata, the better, especially when you start to try to do kind of connect different types of data sets together, having things be interoperable and kind of have some sort of baseline schema for what you're working with is really important for data science, I think. Yeah.
Anything to add on your end?
I think it's also important to note that the metadata is also a hook to other data that is out there, right? So if you look at specimens, but yes, you want to have this one specimen, then you know where it came from and all these things. But once you have that information, right, now you can go, say, out on the internet, right? So you can use the big data analytics, whatever methods, to search actually what is out on the internet around there. And then ideally, sort of in my dreams, right, I would have then something that can, say, digest all the scientific literature about that specific specimen and answer a question that I might have, right? And so the more metadata I have, the better I will be able to embed that one specimen that I have into this whole context.
So all three of you are working on the emerging end, on the edge of practice for your organizations, I would make a guess. And how do you work, or how is this received? How do you get this beyond your kind of department, or how do you make sure that other people understand it? Or maybe if each one of you can kind of talk about how these new practices, and I know that it shifts because just a little anecdote, as you know, this is the reboot. So when we first talked together, Meghan was 3D printing all the time. That's all she talked about. And then we got on the phone for this time round, and she was talking all about VR. Yeah. And then she looked back at the presentation that she had made, like, oh, that's what I was looking at 6 months ago. Yeah. And so we move, our areas move quickly, and it's new. Data scientist probably wasn't a role for that long within the Smithsonian, and I'm sure that you're on the edge of practice within you're Virginia Tech, so maybe you're it's a little different for you. But how do you communicate with other units, or how is it received, or how do you kind of see yourself as a whole, part of each one of you in different ways?
Okay, well, if I can start there, I gave a talk at GW's business school on Monday, and I had about disruptive technologies and 3D printing, and I'd given the same talk a year ago, and I had like completely new slides. It was completely different, and I think that's the key, is that things are moving really fast. In 2013, 3D printing was just starting to come into the mainstream, and we've been doing it since 2007, but it had started to become affordable for the average person, and of course that's exploded. So we were kind of on the leading edge of that. And then it was kind of like, all right, well, you guys have done your thing. And then now that we're getting into the VR and really, like I said, embracing that whole spectrum, it helps when you have Bill Gates come and visit, and then there's like an article about it, and everyone's excited, and now all I'm doing is going and doing a show and tell in VR. But I think that's the key, is like to be able to stay ahead of things. And for our group, because we look for the best tools and techniques to implement for the researchers, that's kind of the key. But again, I wouldn't want to do it if there wasn't scientific value in it, and they've really latched onto it beyond the wow factor.
Oh, that's great.
Yeah, so data science is moving really fast. I think I am, myself and Rebecca, who got hired at the same time, was probably the first data scientist at Smithsonian, I'm pretty sure. But for us, it's really important to get out there and to meet other people in either the museum community or the research community, be able to come and give talks and talk to people. We also live in an age of social media, and so it's helpful to kind of see the pulse of what's going on through places like Twitter and things like that. But really, we just recently published a paper, and so even though there's a lot of criticisms of the slow peer review process, that's also another good way to get your stuff out there, is to go through the traditional scientific publication route. Yeah.
Yeah, so I cannot say that adoption of digitized museum specimens in engineering that is taking off or moving very fast. I have the impression it is a slow process. But I believe at Virginia Tech in engineering, I was probably the first person to show up with data from the Smithsonian. And but the number of people is growing, right? And so there's more of us coming up here and scanning specimens. And I think what helps is obviously that you show your result and you show, look here, I went to the Smithsonian, I got that data. Now I can do something useful. Then somebody else will think, oh yeah, for my research, or people ask me. So do you think I'm studying air-water transition, right? And say, yeah, go to the Smithsonian, they have diving birds, and you can take a look at what their build shape is and then get something out of that. And that actually happened. So I think word of mouth, that helps. And then, of course, being at the university, I would say education, right? So it's really, one thing is to convince people with ingrained habits to go new ways, and that is a very laborious process, but sort of having undergraduate students and telling them about the interdisciplinary links that they can make, I think that is a lot, sort of gives you a lot better return on investment.
Great. Well, we're going to open up to questions from all of you now. Don't be shy. There must be a question. We saw molecules. We saw people walk, not people, Bill Gates walking through molecules. Ha, yes.
I've got a question for Paul about just thinking about machine learning and how it was used for the botany collections to kind of complete the metadata and learn about the collection itself, right? And fill in that information. Do you see machine learning being used on collections in a way that generates something new? Not saying that obviously like this is data that was incomplete, but rather creating something new. And I'm thinking about the keynote from this morning as well.
Yeah, no, that's a great question, and I think that's probably the most exciting thing about machine learning and those different methods that you can use. There's, everything that we did falls under the umbrella of supervised machine learning. So you have an algorithm and you tell it what you're looking for. But there's this whole other area of machine learning where basically you find patterns in the data without having trained anything to recognize those patterns. And so you can feed it data and look for patterns. And I have some experience with those methods, and I think that's a really exciting way to look at the collections, especially for curators and folks, research scientists who are always looking for, new insights into the organisms that they're studying. I think that could be really powerful. So yeah, it's important, and I think that's going to be a big part of the future.
I can just add to that with regard to finding patterns in data. So there's a lot, I'm gonna look at Michael Weinberg because he gets to do with the IP stuff. But in terms of people when they reuse content and when they remix it, and how do you differentiate like when you're adding value and not just, when you get to a new object. But essentially, the community has thought about, well, how can we compare two different 3D models and have kind of like a way to do a diff? So to take a whole repository and then say, okay, well, actually, you've published this with whatever, but it's identical to this model, and this model came before. So I think that's, as far as collections and finding patterns, I think that would be a useful tool, ultimately. And I think there's a group working on that, but I don't know if it's out there.
Looks like we have a question in the middle here.
I suspect that the answer to this is we're working on it and it's hard. But when you mentioned, you're thinking about scanning and figuring out the metadata that you want, and there's a big leap forward from simply geometry to geometry and color information, and geometry and color and attribution information. But then when offhandedly you said, okay, if I was studying things that had to do with entering the water, and I wanted to go to the Smithsonian and find a bunch of birds that dive in the water, if we had scanned those birds, we probably today wouldn't have included information about how the kind of the feathers of the birds react to water and have like additional material properties in that. So as you think about scanning, as you think about building these data sets for the collection, there's the kind of current challenge of building a data set that includes all the information that's easy to use with today's technology. And then there's this sort of perpetual challenge of trying to forecast into the future what other information is it practical and would it ever be useful to include in that metadata set? Besides just saying it's a problem that we know about it, is there a kind of a process or a conversation going on about expanding the metadata and the additional information that we're collecting as we're scanning all of these pieces from these massive collections?
I can add to that, but not with regard to scanning, but with regard to medical models, and how we are bringing more of those into the site and incorporating that. We've talked to a lot of clinicians, and I'm on a working group with the Radiological Society of North America to figure out the best way to organize those. And Paul mentioned schemas and thinking about, but of course, how much do you add? When does it stop? How much is useful? And then, of course, at what point does it just become a pain to have that many fields in a database? Of course, you can add all you want in a header, but yeah, that's one thing that we have been concerned about. So what kind of diagnostic taxonomies can we bring in? So like on our site for heart models, you can search, you can actually drill down this hierarchy of diagnostic codes. So
it sounds like one of the things that you did was you pulled together advisory teams to really forecast, like, or do some forecasting of what do you think would be useful, and if you bring together different people that are thinking about that helped kind of build or at least help mitigate that future proofing again.
And actually, I've been trying to kind of like grab at them to say, well, what do you want? You know, like we can put it in there and then think about in terms of how the data is stored and then the user interface when you're entering it and querying it. But they're like, oh, well, you know, less is more. You know, we don't need that many. And you say, okay, but it's going to be annoying when you come back to me and we have 1,000 things in the collection, and then how do we go back and add that? But it won't be annoying. I'd be happy to do it, but it'd be extra work.
Yeah, so one thing I would add is something that Rolf alluded to is that once you have some type of metadata, you have this awesome ability to go out and link it with other types of metadata, right? And so this idea of linked data, and I mean, I've sat there in committees and labored over metadata schema and stuff like that too. But you know, if you have a taxon name, then chances are you can do a literature search or do things to link it to other databases to pull in more information. And I think since we all specialize in different things, that seems to me to be the best way forward, is to strengthen those ways of kind of exploring other people's metadata to be able to link them all together for the question at hand.
I really like the question, particularly the aspect, right, that once you have a specimen, right, what should you get from that? Right? Should it be just a photo? Do you want to do a 3D scan with color? How about spectroscopy or things like that? And so from my conversations with the people at the National History Museum here, it seems really that the process that is the most labor intensive is actually to get the specimen out of the collection to where you can scan it. And based on that, my advice would be, if you can ever do it, once you have the specimen out, do as much with it as you can possibly do, right? So any machine that you can now think of, right, if you set up for yourself a nice workflow, it shouldn't really matter all that much. But from a scientific point of view, from using this data, that would be the way to go. Anything you get the specimen out, if you have it, do any possible scan, any possible data that you can get before you put it back, because that's really where the work is, and that is your best way, right, to anticipate future needs or see what people would come about. The data, I mean, you see that also in the development, right, of the industry, right, that now the companies, right, that are the most valuable are the ones that have data, right? So data is really a driver here, and so the more you can get, the better, right? And also the more different types, right? So not just how many bytes and bits, but what type of data can you get? So I mean, if you can get it all. That would be my advice.
So it sounds like that was kind of 3 methods. One, kind of finding, like, sitting down with the researchers or end users, basically, and doing more how might we, what if kind of things with advisors. Then we have getting as much as you can, but making sure that you have linking out. And then from our researcher here, get it all, one way or another. So I think that might be a good way to wrap that up, because I think it's great, 3 great opinions on that, and really want to thank you all for sharing all of your stories. Thanks.
Thanks.
Thank you.